Today I would like to tell you about our two main products. They are based on an anti-inflammatory small molecule we call PPC001. One is an intravitreal administration we call Panoptes for non-infectious uveitis. The other one is a nanocarrier-based eye drop, Panoptes. In short, we currently are running a clinical study in uveitis, and we expect to file IND for a dry eye product this year. So, in inflammatory eye diseases, we have a common denominator very often. So, it's very often T-cell driven in dry eye, but also in uveitis. Th1, Th17 cells secrete interferon gamma and IL-17, which leads then to inflammatory processes and then to tissue destruction. What PPC001 does, it reduces the proliferation of these particular T cells and stops the secretion of the cytokines, and therefore no tissue destruction. And in doing that, it's very much unique in addressing the mechanisms of these diseases. So, first, our dry eye product, Panoptes. I don't need to tell you that it's a big problem in dry eye, but what we need to focus on, we only serve 1.5 million out of the 30, 40, 50 million of patients. And we believe that with our clearly differentiated product here, we can support additional patient need. So how do we do that? Panoptes is a unique formulation of an anti-inflammatory, of a new chemical entity, and we use human serum albumin as a nanocarrier. Human serum albumin is known as a natural tear component that is loaded with PPC001, multiple molecules of PPC001 per molecule. Then we deliver it in a normal eye drop. And what we know is that we have on the cornea and on target tissues, we have albumin receptors, which then supports the residence time on the ocular surface. And what we also have shown that it actually gets also delivered, not only loaded, it also gets delivered. So what we have here in our hands is a unique delivery tool with a novel an NZ delivering to the target tissues. We've shown that also in animal model for dry eye disease, we could reduce the fluorescent staining and also inflammatory lesions in lacrimal gland, improve histology. So we get delivery where we need to have delivery to. So switching, so overall, it's clearly differentiated. As I said, it's a delivery tool plus an NCE, quite different from, in the mode of action, very much different from what's out there in the anti-inflammatory, anti-dry eye space. And at the same time, it's highly tolerable. And we've shown in a very small proof of concept study that we are quite superior what's out there. So switching gears now, our second product, uveitis, it's an orphan disease and it's an unmet medical need. And currently it's focused on, the treatment is focused on steroids, and we need to have a replacement for that. And what we have here is an intravitreal injection. We use Panoptes, we call Panoptes, and that is a once every 3 to 4 months injection. And we address the sweet spot in treating locally, not systemically, as compared to other molecules in the field. We've shown that in animal models that it works. The red line shows you the untreated red uveitis, orange is treated with Panoptes. We downregulate interferon gamma and IL-17 and also have the retina intact. So currently we have a 1P2A study running. It's a multi-center study in non-infectious uveitis. It's prospective, open label in several countries in Europe. The objective is clearly safety, tolerability initially, but also improvement, information, and visual acuity. What we can say, since it's an open label study, what we know, the low and medium doses already shown very nice tolerability, so no adverse events, and initial improvement of treatment response. So we're very excited about that. We hope to get final data out this summer, and we are continuing that process. As I said, it's different from what's out there. We don't have any impact on IOP. It's a disease-specific mode of action addressing the underlying cause. It's a local injection and every 2 to 4 times a year. So we have, in summary, to conclude, we have 2 products. One is in uveitis, it's Panoptes. We have top-line data this summer. The second product is in dry eye with a unique delivery tool with a new molecule, anticipating IND this year and data out next year. At this point, I would like to thank being the last in that session. Thank you.